Okay, here's our review podcast for Unit 3, which is genetics. Okay, so we're going to start by taking a look at what nucleic acids are, because nucleic acids are all about genetics. So nucleic acids are organic compounds that either store or transmit genetic information. And remember, organic, the word organic means contains carbon. Let's not forget that, people. Okay, so organic compound that's going to store and transmit our genetic information. The two types of nucleic acids are DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. So DNA has this double helix structure to it. It's a large molecule, whereas RNA is a much smaller molecule and has only a single strand. Nitrogenous or nucleotides are the monomer of a nucleic acid, and they all have the same basic structure. So you have your phosphate group, you have a 5-carbon sugar, which is either ribose in RNA or deoxyribose in DNA, and then you have one of 5 different nitrogenous bases. So the 5-carbon sugar and the phosphate group. In DNA, that 5-carbon sugar is deoxyribose. That's why you have the D. RNA, it is ribose. And remember, sugars all end in OSE, so we know that they are sugars. All right. And then you also have your phosphate group, which is a phosphorus surrounded by a bunch of oxygens. The nitrogenous bases come in 2 groups. You have your purines, which are adenine and guanine, A and G. And then you have your pyrimidines, which are cytosine, thymine, which is only found in DNA, and uracil, which is only found in RNA. So when you put a DNA molecule together, the phosphate group is attached to the sugar, and the sugar is attached to one of the different bases that we have. Now DNA will take on this structure called the double helix, and that is because it's a 2-stranded structure, and they are held together. Those 2 strands are held together in the middle by the nitrogenous bases. And what you can see here when you look at this is like this is a C, and across from it, the purple is a G. And C and G always go together everywhere. And then here you have an A, and the red is T, and it's always A and T that go together. Okay, so that DNA sequence that we get is going to be used as our genetic code. And in the genetic code, we take our little piece of DNA, and eventually we're going to get to the point where we're going to make an amino acid sequence. And that amino acid sequence will become our new protein. So the whole purpose of DNA is to tell our cells which proteins to make. So the DNA holds the code. The code, we actually read it in 3s, which are sets called codons, and we end up with our amino acid sequence, which will eventually be our protein. Now, sometimes we need to make copies of our DNA because DNA, remember, holds our genetic code. So DNA will make a copy of itself, replicate itself before a cell will divide. And that is to ensure that all the cells in an organism have the same DNA. So remember, this is going to happen during S phase of the cell cycle, because S stands for synthesis. So during S phase, DNA is going to replicate itself, and this only will happen before cell division. You don't want your DNA to replicate itself if the cell is not going to be dividing. So when DNA makes a copy of itself, it actually ends up making a mixture of old and new. So here's your original strand over here. Okay, so here's strand 1 and there's strand 2. Those 2 strands are going to split apart, and then new bases are going to come in, and based on our complementary base pairing that we talked about, remember, A goes with T and G only goes with C. Okay, the new bases are going to attach to the old bases. So here's strand 1 again, and here's strand 2 over here, and now they have a set of new bases attached to them. But these 2 sides, these 2 pieces of DNA are identical to each other in the end. Now, another molecule that is going to help with the process of making proteins is RNA. Okay, RNA is a small nucleic acid, and it helps DNA to make proteins. DNA, remember, is too big to leave the nucleus. It's a very, very large molecule, so RNA is going to help DNA get the message over to the ribosomes. And remember, ribosomes are where proteins are made. So RNA is kind of its little messenger. The way RNA gets made is through a process called transcription. So in transcription, DNA is going to get copied into a complementary strand of RNA. This happens inside the nucleus. Okay, and here is a graphic showing that process. I'll let it restart here for a second. So here your DNA molecule and just made an RNA molecule from the DNA. So you start out with your original DNA molecule, an enzyme comes over called RNA polymerase. The new RNA molecules come in. These are the nucleotides for RNA. And notice that instead of T's, there's U's because of the difference. And you get your new RNA strand that gets made in the end. And then the DNA goes back together, and that much smaller RNA strand can now leave the nucleus. There are 3 types of RNA that all have important roles in the process of protein synthesis. The first is called messenger RNA, or mRNA, and that's the one that's going to take the message from the nucleus, the DNA in the nucleus, takes that information and brings it to the ribosome. Okay, so that message goes from the nucleus out to the ribosome. Ribosomal RNA is what ribosomes are actually made of. So this is going to make up your ribosomes. And then the third kind is transfer RNA. Transfer RNA is actually, its job is to transfer or carry amino acids. And remember, amino acids are used for making proteins. And that is going to happen at the ribosome. So one of the things, again, the MCAS people like for you to be able to do is to compare DNA and RNA in terms of their structure. So DNA is a double-stranded molecule, quite large. Its sugar is deoxyribose, that's why it's got the D. And it has a base called thymine in it, which RNA does not have. So when you look at RNA, RNA is a small single-stranded molecule, so it's only one strand, so here's your RNA down here. It has ribose as its sugar instead of deoxyribose. And instead of thymine, RNA uses a base called uracil. And those are the biggest differences between DNA and RNA. Now, DNA and RNA are going to work together in order to do the process of protein synthesis. This is a multi-step process. It starts with transcription. Okay, so we have genes, and our genes tell us what proteins to make. So a gene for whatever protein we need to have gets transcribed into a piece of mRNA. So we're going to make our little piece of mRNA. The mRNA is going to take that message, leave the nucleus, and go over to a ribosome out in the cytoplasm. And then the process of translation is going to happen based on the genetic code, and that's when we read the DNA in threes, like TGC. Okay, the mRNA is going to bring over specific tRNA molecules, and those are carrying the different amino acids. So each tRNA molecule is going to carry one of the 20 different amino acids. They're going to get attached based on the genetic code, and eventually you will make your entire new protein. Okay, so there's DNA in the nucleus, too big. So we make a piece of RNA that can get out, which goes to the ribosome, and then you eventually will get your protein chain that gets made. Okay, here's another picture of that. Here's DNA. So this piece of DNA has multiple genes on it. So if we only want to make this particular gene, then we're just going to take that piece of DNA, transcribe it into this piece of RNA, and again, complementary, A goes with U, C goes with G. And then we read it in threes, and each of those threes corresponds to one of the 20 different amino acids, and that makes up our protein chain in the end. Okay, so DNA gets transcribed into mRNA, mRNA gets translated into a protein. The transcription piece happens in the nucleus of the cell because remember DNA is stuck there, it's too big to get out. And the translation piece happens at the ribosomes because ribosomes are where proteins are made. Now, every once in a while, a mistake will get made in your DNA, and you can change the sequence of nucleotides in a gene, and that's called a mutation. And what can happen is you can get a completely different protein made. Sometimes an entire chromosome can be changed, and that can cause genes to either be missing or you can have extra copies of genes or extra pieces of genes. Any of these could have bad effects, or they could actually have good effects in terms of natural selection. The most common types of mutations that happen are point mutations, where you're going to have a change in one letter. So here you can see that you had a G in the RNA and it got changed to an A, and that ends up changing this amino acid from arginine to histine. So we change one little amino acid. Same thing happens here if you remove one amino acid. You add an amino acid in, it will completely change the sequence from wherever that mutation happens. And if you delete something, remember this one, we had a sentence, the fat cat ate the rat, and if you take that H out and then try to read it in words of 3, you can't read it anymore. It's no longer makes any sense. So mutations can have pretty drastic effects on different proteins. The least substantial of those is the substitution, but you can still get some pretty serious disorders that happen because of substitutions, like sickle cell anemia as an example. Okay, so all of this is going to lead to our genetics. Genetics is where we're going to study heredity. Okay, and remember, heredity is how you are going to get your traits. Traits are caused by the genes that you have. A gene is a piece of DNA, and that's going to determine whatever protein the cell is going to make. And those proteins are what are going to determine your traits. Okay, so your genes determine what you look like by deciding the proteins your cells will make. So if you have brown eyes, then you made a brown protein that gave that pigment to your eyes. Okay. Now for all of our genes, we have different alleles. So a gene can have 2 different forms called alleles. And you have 2 alleles for every gene, because remember, you got one set of your genetics from your mom and the other set from your dad. So you have 2 alleles for every gene. Alleles tend to either be dominant or recessive. Okay. And we will represent our alleles by using letters. So if something is a dominant allele, that means it will show if present. And we represent our dominant alleles by using capital letters. So anything that's a capital letter means dominant, and the recessive is the same letter but in lowercase. Okay. So when we talk about our pairs of alleles that we got, if you have 2 alleles that are the same, these are called homozygous, because remember, homo means same. And we also call this purebred or true breeding. So the genes that come together in the zygote with the same alleles, that means the allele is the same. So you can hear this person, this P here is big Y, or little y. Okay. So for the example of tall, somebody who's homozygous would be big T, or if they're short, little t. That's different than heterozygous. Hetero means different. Okay, so the other word for this is hybrid. Think of a hybrid car that's both gas and electric. These are genes that come together in the zygote and they have different alleles. So instead of them both being capital or both being lowercase, you have one of each. And when that happens, the dominant allele is the one that is shown. So big T, little t is tall, even though it's got one dominant and one recessive. You don't get the mix of it. It's not medium, it's tall. Okay, some other words that are really important when we talk about genes. Genotype is a genetic makeup of an individual. We represent that using letters. Okay, these are the genes that a person possesses, and again, we use letters to do that. So a genotype could be big T. That's a genotype. Phenotype is the physical characteristic, what something looks like. Okay, so remember, pheno, physical. Big T and big T, little t are tall. Their phenotype is tall. Little t is the genotype, and its phenotype is short. All right, so Gregor Mendel was the person that did a whole bunch of experiments in the 1800s on genetics, and he came up with 4 fundamental laws of genetics. The first is biological inheritance, and that basically says that traits get passed from parents to offspring. We now know that those things that are getting passed on are through genes. The law of dominance. When 2 forms of the gene exist, remember we just called those alleles, okay? One may be dominant over the other one that we call recessive. His law of segregation. So alleles will segregate from each other when you make your gametes. So remember, your gametes are your sex cells, sperm or egg. So when you go to make your gametes and you're going to give your DNA to your offspring, you're only going to give them one of the 2 alleles that you have for each gene. You're not going to give them both. And his third law was independent assortment. And this says that entire chromosomes and the alleles that are on them separate independently of each other when gametes are formed. So you have 46 total chromosomes. The law of independent assortment says you're only going to give 23 of those chromosomes to your offspring. Now, what we can do when we are talking about genetics is we can actually do problems using a Punnett square. A Punnett square is going to predict the gene outcomes for a genetic cross. Okay, so we can predict what the genotypes and phenotypes may be for the offspring of a particular mating. It does not mean that that's what's always going to happen. So if you were going to draw a Punnett square, okay, and this is a monohybrid cross, so we're just going to look at one trait at a time. So let's say we're going to cross big T, little t, little t, little t. So remember, you take one of the parents and put them across the top, and you put the other parent down the side, and you get big T, little t, little t, little t, big T, little t, little t, little t. Okay. So if we were to do our phenotype and genotype ratios, our genotype ratio would be 2 big T, little t, to, oops, we don't want that. It should be a colon, to 2 little t, little t. Our phenotype ratio would be 2 tall, because big T are tall, to 2 short. And that would be our expected outcome if we crossed these two parents right here. Sometimes we can make Punnett squares that are much bigger that show independent assortment, so you're looking at two different traits at once, but we'll keep it nice and simple here. Now, there are exceptions to the rules of dominance. So sometimes things are not, one allele does not completely dominate over another one. So the heterozygous individual ends up being a mix of the two homozygous phenotypes. So here we have a white and a red. And if you mix white and red together, big R and little r, you end up with pink. That is the mixture of the two. Where normally, in normal dominance, big R, little r would still be red. So in incomplete dominance, they're kind of blending together. Okay, so big R is red, little r is white. So red is dominant to white, but it's not completely dominant to white. So when they mix together, you get pink. Codominance is similar, but in this case, both alleles are going to contribute to the phenotype. Both of them are dominant. Very often in codominance, there is no recessive allele. It's just both of them are dominant together. So the heterozygous phenotype will end up showing two traits distinctly. So a good example of this are the black and white feathers on this chicken. This chicken's got both black feathers and white feathers. They're not blended together. If they were blended together, the chicken would be gray. But because it's codominance, it has both black and white feathers. Okay, multiple alleles is another variation in genetics, and this is where you can have a gene that has more than 2 alleles. Okay, normally we've been talking about genes, you either have, there's a dominant and a recessive. In this case, you can have multiple things. So this increases the variety of phenotypes for a population. So we talked about blood types. There's a lot of possible genes. You can be little I, which is the recessive. You can be big A, big B, okay? So there's 3 different possible alleles for blood type instead of the 2 that we normally talk about. So instead of just getting 2 or 3 different phenotypes, you actually get 4 different phenotypes. So you have more phenotypes possible. You also get a variety of phenotypes when you're looking at polygenic traits. These are traits that are controlled by 2 or more genes, and you get a very wide range of phenotypes when this happens. So if you think about human height, some people are at the extreme short end, very few people are at the extreme high end. Most of us fall somewhere within the middle range, and that would be average. So the average height for people is 5.5 feet, so 5.5 feet, okay? That's like 5'6". Next, on either side of that, you have your 5 feet and you have your 6 feet. Most people fall in that range, and then it's only the other extremes, and that's because there's many genes that will control the height of human beings. And the last thing we talked about is the difference between our sex chromosomes and our autosomes. So combination of sex chromosomes from the gametes determines the sex of an individual. So remember, you have 22 autosomes, and then you have a sex chromosome. The female parent can only give the X chromosome, that's one of your sex chromosomes. The male parent can give the X or the Y. So depending on what the dad gives, you're going to have a boy or a girl. So mom's always going to give the X, but dad, if he gives his X, those are your girls. If he gives his Y, you have your boys. Now some traits are inherited on the X chromosome. When males get the X chromosome that has a trait on it, they're going to show whatever that trait is because they only have one X chromosome. So whatever is on that X chromosome, they're going to show it. Females have two X chromosomes. Okay, so they have two copies of every gene. So dominance may be a factor for them. They may not show a recessive trait because they have two copies of the gene. It's a little bit different. Okay, so here you have a female that's heterozygous. Okay, and here you have a male that's just dominant, whatever. So if mom gives this one over to her son, it's a male unaffected. If she gives the blue one, that's the male affected for whatever this trait is. Let's say color blindness is an example. Whereas the females, because dad has the blue one here, you can actually have a female that ends up with both blue ones, or you can have one that has one of each. Okay, but the girls don't necessarily show it. The boys are going to show whatever is on that X chromosome because they only have the one, whereas the girls have a chance of overpowering one that shouldn't be there.